In this presentation, we're going to go over the third part of kind of the process here. If you remember, we kind of left it with the electrical signal, the action potential being sent down the axon of the neuron. And if you look at that picture that is the synapse, you can see that the electricity comes down, but the electricity of the action potential doesn't go across the membrane. It doesn't go across the synapse. It's actually neurotransmitters that do that. So we have to go over something called neurochemistry, which is kind of the action of how neurotransmitters are made, stored, released, and do all their funky things. So your outline actually has a number of the processes involved, so let me share that with you, and we'll go through it. The first step, and the question I often get is, can you actually just eat a neurotransmitter? Can you actually just eat serotonin? Well, the answer is yes, you actually could just eat a neurotransmitter. The problem is it would not get through your blood-brain barrier, which is a thin layer of just epithelial cells that are lined up against your neuronal membranes as they go through your brain. So all the neurotransmitters, and you can see the list: serotonin, acetylcholine, dopamine, norepinephrine, epinephrine. Yes, you could eat them, but they wouldn't get through the blood-brain barrier, so they would not be synthesized. The first step is you have to synthesize neurotransmitters, and as the outline shows you, the picture, basically your diet, things you eat in your diet are converted into neurotransmitters. They are synthesized from precursors, things in your diet, and enzymes and so forth. They are taken back up in the cells, and they are converted into neurotransmitters. So step one, you've got to make the neurotransmitter. Step number two, as the drawing says, these neurotransmitters are stored basically right at the end of the axon. They're stored in the presynaptic region, and they're stored in structures called vesicles, which are basically little packs. It's certainly not the case that if you have a neurotransmitter that it's just kind of floating around and it's all released at once. You have a lot of neurotransmitters up in your brain. They're all stored kind of right at the edge of the presynaptic region, and only a little bit is released at a time. So step number two is the neurotransmitter is stored. Step number three, not following the outline. We're going to ignore what it says there with leaking and so forth. Step number three is really the critical step. It's actually called step 4 on your picture. It's the neurotransmitters are being released, and what basically happens is the electricity from the action potential comes down to the very end of the axon, and it'll cause the vesicle to fuse with the membrane and release the neurotransmitter. And there's a picture on the next page. It's called exocytosis. I guess it's right above the word exocytosis, which kind of shows the vesicle fusing with the membrane, and then it releases the neurotransmitter into the synapse. You actually need to have electricity there, as well as a few other elements as well. So step 3 is the release of the neurotransmitter, and this is where the electrical story maps onto the neurochemistry story. So the neurotransmitter is released. The next step, which is actually called 6 in your picture, is the neurotransmitter binds across the synapse. It binds at the postsynaptic receptor. That is the postsynaptic receptor, which is the dendrite of the next neuron. So it binds there, and after it binds there, it's going to actually open up a channel. It's going to open up an electrical channel, and if it works out right, another action potential will happen in the next neuron. And then the very last step is actually called inactivation. After the neurotransmitter does its thing, basically a neurotransmitter can then be destroyed by an enzyme, or the other way that a neurotransmitter can be deactivated is if it's kind of recycled and put back up into the presynaptic region. So those are the two ways to inactivate a neurotransmitter. And finally, I do want to share a little more detail on information about the binding of the neurotransmitter here. If you look at this page after it says the word excitosis, there are basically two mechanisms by which binding leads to another action potential. One of those mechanisms is where the neurotransmitter binds at the channel and simply opens up a protein channel that lets an ion in. So let's say you're the neurotransmitter glutamate. When glutamate binds, a channel is open to allow calcium to move. And calcium is labeled Ca++, and calcium, when glutamate binds, rushes in to the postsynaptic region, and because calcium is a positively charged ion, it causes a positive electrical charge. Sometimes those are called, remember, depolarization before, but now since it's after the synapse, it's called an excitatory postsynaptic potential, and that will cause another action potential to happen in the next neuron. So the neurotransmitter binds and then opens up an ion channel. The other way a neurotransmitter can work is called via a metabotropic mechanism. The other one we just covered was ionotropic. In a metabotropic mechanism, the neurotransmitter binds at the postsynaptic receptor, and it'll basically send a second messenger to another spot and then open up an ion channel. This mechanism will take a little bit longer to occur because the second messenger, in this case cyclic AMP, takes a little more time to travel, but when it does open up the channel, it's going to open it up for a longer period of time. So the way a neurotransmitter binds and opens up the channel can either be via ionotropic mechanism or metabotropic mechanism. And to continue the last part of this presentation, inactivation. I want to talk just a little bit more about that. Many of you are familiar with drugs that actually help with depression. And the one example I will give you is a drug such as Prozac. There's Prozac, there's Paxil, there's Zoloft, there's Lexapro. What those products do is those products are called SSRIs. And what they do, what SSRI stands for, is selective serotonin reuptake inhibitors. What Prozac and the other products do is they block the reuptake of serotonin. They block the inactivation of serotonin. They'll block the reuptake. They'll keep serotonin in there longer. And so they're actually helping to increase serotonin functionality. And when you have more serotonin working, you tend to have less depression. So hopefully this makes good sense to you. If you need to look in your book or type in some notes and figure some more things out, go for it. Just try to put your outline together and give it your best shot. Thanks. Bye-bye.